Hello, everyone, and welcome to today's webinar, Tilting Against Windmills, Clarity in Spectral Panel Design Without Losing the Battle for Biological Resolution. I am Matt Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Seddon Thomas, Staff Scientist, Thermo Fisher Scientific. Seddon, you may now begin your presentation.
All right, thanks so much. So the title of the talk is, again, Tilting Against Windmills, Clarity in Spectral Panel Design Without Losing the Battle for Biological Resolution. And just what I'm referring to, this idea of tilting against windmills comes from Don Quixote, and the idea that instead of fighting a monster, as he thought, he was fighting a windmill. And since a windmill would continue to turn, no matter what, it was always going to be a losing battle in terms of that. And so it's a really good analogy just to start thinking about spectral panel design. So the problem with building to higher parameters is both the limit of the detectors available for the unmixing, as well as the increasing spread. And so, you really want to look at what dyes you're using in terms of utilizing this underutilized space. So when you look at a spectral panel, there's a lot of spots that are really not used. And so we also are trying to use lessons learned from previous 45 color panel design to really drive that new panel design. And the goal is really to understand how pushing towards a theoretical maximum parameters affects panel design and biological resolution. And one of my goals is to make this clear so that you, instead of fighting against the way something works, you're working with that and using that to help solve the problem as you build towards higher parameters in your panel. And so, the idea here is that when you think about hardware innovation, it's really driving die development. So back when there was really just a 4-color fax caliber, there really was very little pressure to have more dies. As you start to have more sophisticated cytometers going to 12, 18 colors, and then pushing into spectral, you really move to this pressure point where the high parameters puts pressure on detecting similar fluorescent labels and accommodating non-ideal dye performance. And so, as you can see, there's just this continued increase in terms of what the dye, how the dyes need to perform. And so when we think about, conventional cytometry versus spectral cytometry, I think this is one place for us to really understand what's going on. And so I have here a dye, APC, and you can see that, you have your excitation curve in this dotted line, and then you have your emission curve. And so APC is predominantly excited off of the red laser. You can see here from the excitation curve that it would also be predictably excited off of the yellow-green laser, but not so much off of the rest of the lasers. However, when you look at a spectral signature, what you can see is it tells us a little bit different set of information. So of course you do see the fluorescence coming off of the red laser, and that is the primary emission spot. And you do see this emission coming from the excitation off the yellow-green laser. But one of the things that you don't necessarily capture is the fact that there is this emission also coming off of the violet and the UV. And so sometimes what spectral cytometry does is really show us what is happening even on a conventional instrument. And so, if you've used a number of dyes and start to notice, hey, this is, affecting some of the other detectors, spectral cytometry is just showing us, hey, this is predicted actually based on this dye. And so we can really see what's happening, in your overall panel. And this is happening whether you're using a conventional or a spectral instrument. And so this is just showing you, again, what we would think of as the off-target emission spots in terms of that cross-excitation. And so, when you look in spectral cytometry, you have a dye signature, and it's really the result of non-ideal dye performance used to great effect for phenotyping. And so you can see here up top, you have APC, and at bottom, you have Autofluorescence 647. And although on this aurora, you can see that APC is primarily detected in R1 and Autofluorescence 647 is primarily detected in R2. The real difference when you look at a signature is not so much what's happening in the primary channel, but some of the differences that are happening perhaps in the violet or the UV, because it's this entire signature that's being looked at. And so, understanding that really helps you both in your panel design and increase the theoretical limit of the colors in the panel. And so one of the things we tried to think about on the dye side was, how does underlying conventional dye performance affect your panel design? And conventional dyes can only perform in two ways, and both can lead to spread or loss of data resolution. And so in this slide, you can see here we have something like brilliant violet 510, which is great in that it's not cross-excited, but this broad emission that takes up a large number of channels in the violet spectrum still can be somewhat problematic as you start to detect more and more dyes. And then there's other dyes like PE Dazzle, and Percy-P that are cross-excited by both the blue and the yellow-green laser. And again, those are taking up spots. So if you had something that was excited off of only one of the lasers, it's taking up less space in your panel. Additionally, when you think about spillover and data resolution, here's an example from the University of Chicago blog. And you can see here that it's showing the plots for APC size 7 and PE size 7, and you can see that in terms of their emission, although they're excited primarily by different lasers, that their emission is also is in a similar spot. And so just as an example here, this is showing you if you had a single color control with APC size 7 and use this uncompensated, and then the idea of if you applied compensation. And in an ideal setting, you would imagine that this would line up perfectly. You would have no, detection in PE size 7 from this single color control. But in reality, what you see is this, it looks like either a trumpet or a goldfish tail, this spillover spreading error. And so what this means is that you're losing resolution of your data. So if you had, something that was incredibly bright, you would easily be able to separate it. But if you had something dimmer, it would be harder to determine what is noise and what is your signal. And so, in this context, we would refer to APC size 7 as the perpetrator and PE size 7 as the victim. So basically, you're losing that resolution in your PE size 7 channel because of this. So we stepped back and said, could we really design a better performing dye, and what would that look like? So there's an imaginary dye here, perfect blue, and you can see that that's showing up in one of the 64 detectors off of the aurora. And then we have a tad less than perfect blue. And so perfect blue would be great to have, but it does break some of the laws of physics in that you would have to have some decline in terms of this. It's, not acting just in that one detector. And so the goal would really be to generate something a tad less than perfect blue. So having, a really strong signal, no cross excitation, and a very rapid decline in terms of the other channels. And so that's what we tried to do here. You can see with the Invitrogen Phyton technology, we're taking single-stranded DNA, and then it is then folded into this cruciform structure. And what that allows for is for us to position dyes very specifically on this structure. And so we can actually engineer the excitation and emission. So this is using FRET technology. So using positioning a donor and acceptor dye at specific positions to engineer this fluorescence by design. And then we can attach that to the antibody. We haven't done as much with this, but this does also enable us to tune brightness if we wanted to, instead of putting it on 2 of the arms, put it on more. So in this example, it's just showing you the idea of having a 4-cluster version. So, if you think about what you'd want in terms of dye requirements, you'd really want something to be bright and clean, really to limit laser cross-excitation and minimize spread. You'd want it to be consistent and stable, and ideally have controllable brightness to match based on antigen density in the panel that you're using. And so we tried to take a few different targeted approaches to designing new fluorescent labels. And so I'm going to be talking about this, but we started with the idea of separating out, one of the primary issues with blue and yellow-green excitation as compared to existing PE and PE tandems. And then I'll talk about a number of other things that we've approached in terms of thinking about dye design. So in terms of thinking about your panel, the idea is that PE dazzle, you can see this is an excitation curve rather than an emission curve. And so what that's showing you is that something like PE dazzle is excited primarily, it's excited by the blue laser and the yellow-green laser. Whereas something like NovaFluor Blue 610 is excited primarily by the blue laser and not so much by the yellow laser. And the same for NovaFluor Yellow 610 is excited primarily by the yellow laser and not so much by the blue laser. And so what this does is really allow you to get this 2-for-1 situation where you can actually use 2 NovaFluors in place of just one, previous PE tandem. And of course, this depends on having the filters. So a number of folks that have built cytometers before this have built around this problem, and that's why you tend to see less detectors off the blue laser, but that's not necessarily required. And so the next thing we tried to attack was this problem of building more red-shifted dyes. And so when you think about the goal is really to get something that is a long Stokes shift from a defined primary laser. So here you can see you have your typical donor dye, you have your acceptor dye, and you have high spectral overlap. And so it's very easy to transfer that energy from the donor dye to the acceptor. But when you think about doing this, further down across the spectrum, you can see that then the donor can be excited, but it would just tend to emit where it is because it's too far away from the acceptor. And so thinking about how to solve this problem. And I'm just putting this here from a small subset showing off the yellow, green, and red off of the Aurora. And you can see that there are these large gaps. So between YG5 and 9, there are not so many dyes, and between R4 and R8. So there's these open spots, and this really does relate to building more redshifted dyes in that way. So if you think about this challenge, in this case, the idea, in terms of American football, that, if you're throwing a short distance, it's, not too hard to make that pass. But if you're throwing a really long distance, you require more energy to make that pass. You really need to be very specific in terms of reaching, in this analogy, someone that's going to catch that ball, they're going to have a lot more skill. And so it's a similar idea in terms of this challenge. So you have to go a long distance across the challenge, and cross excitation can be an issue. When you think about this in terms of the dyes, the acceptor is larger and more unstable and thus susceptible to oxidation. So this is definitely something that we're continuing to build towards, and we are in process of building more redshifted dyes that will be released to market. I think as of this past month, we've released NovaFluor Blue 690, and again, we'll be releasing more of these red-shifted dyes in the future off of these laser lines. So when you think about the challenge in terms of switching gears, building out ultraviolet and more violet dyes, which we have not released yet as NovaFluors, UV and violet excited dyes are harder to separate from the background due to the autofluorescence of specific cellular components. And so you can see here, this is again coming from the Aurora, and this is just showing unstained cells, unstained PBMCs. And you can see that there are contributors in terms of autofluorescence off of the UV and violet background. And these can include amino acids, structural proteins, energy pathways. So you're picking that up in these cells. So that means that you have to build your brightness higher to get the separation because you already have some more background noise there. And also, the extinction coefficients are generally lower for smaller UV excited levels that lead to lower energy emission on a per molecule basis. So just calling this out, so the UV dyes that have been released thus far in terms of the brilliant UVs, those are polymers versus single fluors. And so that's one way to build, around this problem in terms of the extinction coefficient, so having to put more dyes on a per molecule basis. And there's also the issue of the use of lower powered lasers for these UV excited dyes, especially on the aurora. And so this is an example which was pretty interesting, I think. So if you look at Q.800, so this, in terms of its excitation, would tend to be excited more by the UV laser than the violet laser. However, the aurora has a 20 milliwatt laser for the UV and a 100 milliwatt laser for the violet, so 5 times as much. So instead of showing up the primary channel in the UV, the primary channel shows up in the violet, and that's really related to the lasers and how this is detected. However, when you look at the Invitrogen flow cytometry panel builder, this is showing you for the 7 laser Bigfoot sorter. You can see that, basically the Q.800 would be predicted to have its primary excitation coming off the UV laser and less off the violet, because these lasers are both 100 milliwatts. So when you're using that equivalency, that's what you have. And so just, taking you into a little more detail here, this is, showing you the instrument configuration for the aurora and the different laser wattages. And, when you think about the aurora versus the Bigfoot, you can see that you have APDs on the aurora versus PMTs on the Bigfoot. The aurora uses the APDs, and so it will tend to detect better in the red and worse off of the UV and the violet, just because of the physics of how that works. And the converse situation in terms of the Bigfoot. So you would tend to detect better in the UV and not as much into the red. And you can also see that there are different wattages of these lasers, which always affects the signature of a dye. So these are the things that we're sort of looking at as we're building new dyes. When you think about then taking all of this information and switching gears in terms of panel design, there are a lot of things that you're dealing with, and you don't necessarily, these problems don't go away just because you're switching from conventional to spectral cytometry. And some of those are thinking about spectral overlap, antigen density. So, do you have something that's bright enough to match for a lower expressed antigen, or something that's a little dimmer for something that's, perhaps, more highly expressed. And then, thinking about the spillover spreading, which I discussed before, and again, matching that staining index to the antigen density. And so this is showing you the NovaFluor dyes that we've released thus far. We've also released NovaFluor Blue 690. But we really haven't released anything into the UV and the violet. And so, we refer to that as purple rain. So I'd say stay tuned. But in the meantime, Thermo Fisher has actually released both antibodies conjugated off of the BUV and the BV in terms of the dyes, the brilliant ultraviolet and brilliant violet dyes. So that's something that you can use in the meantime to really build out your panel more fully. And so we also are continuing to expand the NovaFluor antibody conjugates. So this is with the original 19 dyes. We have all of these clones in all of these dyes. And this is actually out of date. I think we've reached greater than 100 clones at this point in terms of NovaFluor conjugated antibodies. So, if you haven't seen it there before, keep checking the catalog, because the team's been working pretty hard on that release. And we're trying to make sure that we're matching up places where the antibody is bright enough to give the separation that you need for that specific antibody. So we look at all of those issues. So now stepping back and really thinking specifically about spectral panel design. The goal is to build around cross-excitation and spectral spread by selecting spectrally clean dyes and utilizing the spectral distribution in the panel. And so what you can see here, this is just showing you the spectral distribution on Aurora. And at left, you have the different wavelengths approximately that would be there. You have the different channels, and then the different fluors that would be primarily detected in those channels. And so, this is one way to really look at what's happening. And so I think one of the things to draw your attention to is something like BUV 661. So you can see here that it might not necessarily be obvious that you would actually have some cross-excitation off of the red laser, but you can really see that when you have laid out the dyes this way. So I find this layout actually pretty helpful when I'm building, to a larger panel here. And so you can see, that's one way to look at that. So if you had something that might be highly expressed and cause an issue in terms of your panel, you might put something on, say, BUV 395 or 805 or somewhere else so that you weren't dealing with those cross-excitation issues. In addition, as you're building a panel, thinking about, what cells you're putting next to what antigens you're putting next to each other. So, you could put something like CD4 and CD8, but you might, avoid something that would be co-expressed on both those cells because it makes it harder to resolve. And so that brings me to, another question that you should really think about as you're building out your spectral panel. So consider if your panel is meant to address broad or deep immunophenotyping. And so I just want to define those terms quickly. In a broad panel, you really want to position co-expressed markers to avoid spectral spread or cross-excitation. And so an example of this might be looking at multiple lineages in a panel. So T cell, B cell, monocyte, macrophage, those are going to be expressed on different cell types. And so you can actually get around sometimes bad dye performance by positioning these on different cells so they won't be co-expressed. In a deep panel, fewer colors can be used, and you're often dealing with co-expression. So this is showing, for instance, the idea of a T cell only panel. So you might be looking at activated CD4 T cells and then Tregs, and those are going to share a number of markers and then have others where they're not. And so you can never build to as many colors with a deep panel because you really have to deal much more with dye performance than positioning the dyes around, the different cell populations. In addition, panel design is still related to dye antibody clone breadth as well as availability. And so what do I mean by that? So here, if you think about that, if there's, say, your favorite antibody is only available in 3 colors and you've already used up those colors in your panel, you will have to think about a new way, even if you've built a better panel. And then, of course, we've all been dealing in the pandemic with availability issues. So you may have built the best panel in the world, but if you have to wait on that antibody, sometimes you have to switch gears. And so it's very important to think about, how you plan for that in your experiment. I set a goal to build a broad immune panel with spots for selection of fluorophores with the most available antigens. So thinking about FITC or AlexaFluor 488, PE, and APC or AlexaFluor 647. And the idea is that, as we go along in immunology, someone is always discovering the next important molecule that's going to be important for your work. And so leaving a space already built into the panel so that you can actually drop in what you need is actually pretty important to be thinking ahead so that you're ready for that. And so, here you can see this is a panel that I built out, and I put those available spots into the panel and had to then think about, how the dyes perform, even if you have something in there as a placeholder. And so you can see here, for instance, again, to come back to that issue in terms of cross-excitation, what I did so that I could use, this full part is position, IgD, which would be on the B cells, CD1C, and CD3 on T cells. So really trying to position the cells so that they're not co-expressing on these particular fluors. And so the other thing that I I wanted to look at also was the same thing. So looking at positioning, CD4 and CD8. So even though those are both on T cells, positioning them, you can be positioned here because they are still on separate T cell populations. And then looking at other markers like monocyte marker, and sort of separating those out. So making sure that I'm trying to look at different cell types so that you're dealing less with the issues of spread into these channels. And then again, as I mentioned, already leaving these spots available to continue to build. The next thing that you really have to deal with is making sure that your antibodies are titrated for the specific channel and brightness that is sufficient to resolve populations of interest. And this is important no matter what cytometer you're using, but it can also be an issue that maybe an antibody dye pairing is suboptimal. And so in this case, my initial positioning, I was putting CD127 on BUV 496. And so, when I tested that particular conjugate, it really didn't have enough expression, even as I titrated up and gated on the lymphocyte population, as compared with the controls here in BUV 737 or super bright 436. So either using, the same clone or a different clone still really didn't identify that population. So this was something that, after building out the panel, dealing with any, issues of supply, then had to relook at that. And so just as an example here, this change even in a CD127 means that, I can switch that over from here to moving that in place of the super bright 702. And then, because I had built this rather tightly, having to move CD11C to BUV 661, and then moving IGD to FITC in this placeholder space. And so these are the kinds of issues that you typically, need to deal with. So you may have tested your panel on paper, but once you test it, and even do the titration before testing out the panel, this is an issue. And so again, here, there was a place where really needed the brightness for CCR4, so needed to switch to that clone, and then also switch out, have something in here in terms of PD1 and CD86 just to have something to test into. So, now that I've talked about sort of designing this on paper, titrating your antibodies, then really also thinking about selecting dye combinations to minimize the similarity index. And so that's the pairwise measure of the spectral uniqueness, and the complexity score, which is a measure of the difficulty of the panel to unmix and really avoid spectral bunching. So what do I mean by this? So this is showing you the Thermo Fisher panel builder. In this case, it's taking a previously used Aurora panel. And so what you can see here is that you've got, one dye. And so when you get into the darker blue, that's showing you that these two dyes are very similar to each other and thus will not be a good combination. So you can still sometimes use them. It just means that that would be less recommended. But as you build to more and more colors, it's always going to become a bit more of an issue. But interestingly, when you put, say 3 dyes that are in a similar place, there's actually a worse unmixing than you see with any of the individual dyes. And so this is what I mean by spectral bunching. So you actually see that, while you can use this, it leads to worse issues that are captured. And interestingly, when you're building out the panel, you can look in the panel builder at the complexity score. And one of the things I noticed as I was building this was that the complexity score didn't necessarily go up when I added these in a very small panel. And when I talked to a mathematician about this, what they said is that early number was already telling you had made the worst decision possible. And so by adding these 2 additional dyes that were, in the third dye that was in this area, basically it's already saying I've made the worst possible decision, but the complexity score isn't necessarily reflecting that. And so it's just worth being aware of that the complexity score doesn't necessarily capture all of that, nor does the similarity index. So just being aware as you're looking at your panel about that issue. And this also happens for the combination of BB515, FITC, and NovaFluor Blue 530. So what you can see here, again, going back to this panel, is the idea that if you step backwards, you can see places where I could have included more of these dyes. And so what I've done is draw a box showing you that while I could have used all three of these dyes together, in general, that's not going to be your best panel design. And the truth is, you can build to more colors, but if you don't have the biological resolution, it really is a problem. And so in these two cases, as I showed you before, I would tend to avoid using one of those. So you could use two of the three to get better resolution. So that's just worth noting here. And last, as you start to build and test your panel, you really want to assess channel resolution through the spectral spread matrix to avoid remove spread and identify dyes that act as perpetrators and victims. So how do you do that? So if you actually in FlowJo, and I believe it's also available in FCS Express, you can generate this spectral spread matrix. And so what this does is sort of give you a relative indication of the spread. And so when you add this up, so if there's a column of dyes here, you can see when you add this up, the row sum should be able to identify fluorophores that cause a lot of spillover. And this would be then the perpetrators. If you look at the column sum, you can identify fluorophores that have a lot of spillover. And that's important to really understand, where you're going to start seeing the problems in your panel. So those are the places where you might not have enough resolution to be able to, resolve your populations. And so you might need to think at what's contributing to that. And it could be multiple fluors there. So just to be aware of that. And to show you an example of that data, this was, some learning from the initial 45 plus color panel that we tested. I had added in 10 additional NovaFluor dyes, just to see what happened in terms of the spread and the unmixing. And so you can see here, this is a way to evaluate these additional targets. And so when you're looking at, the blue laser, there's very little spread here. And this is showing you, in this case, the column sum. And in this, I had removed the FITC antibody, and you can see you could actually use all of these different NovaFluor dyes together in the panel. And one of the good things about having removed FITC and BB515 as compared to the previous panel is that those were the most similar of the dyes versus the others that I'm showing you here, NovaFluor Blue, 510, 530, 555. So this actually then helped reduce spread. But what you can tell right away is that there's a big issue with spread, as we built more dyes using the red laser excitation. And while I won't go into that, I'll just show you very quickly. You can see that we added a number of red excited dyes. And in general, I would not recommend using APC Alexafluor 647 and NovaRed 660 because they are very similar in that context. So I'll go back. And so this is a way to iteratively really evaluate your panel. So in terms of thinking of additional resources for panel design, Thermo Fisher has a number of options. So, we have the First Panel on Us program. So that's something where you can reach out to get help with designing your panel. So that's there. There are also a lot of really good tools online. So, looking at either for conventional or spectral cytometry, the panels design tools, so you can actually see, where are these spectra, and if you already had a certain number of antibodies in your panel, it's able to deal with that. So you could say, I already have, say, these 10 antibodies, and now I want to add these other 5 in there, and it will provide recommendations. And then you still want to, of course, evaluate that using your own judgment. Is it matching the brightness that you need? Is it, an unusual fluor where you need to really leave space because it's going to be, restricted in some way? Are you having to do a conjugation? Things like that. But a lot of resources online, so I would encourage everyone to check that out. And so, in summary, when you think about, really your spectral panel building goals and validation, it's a really good idea to leave space for fluorophores in the panel to allow for easier incorporation of the rare antigens, as I mentioned. So things like FITC, PE, APC are typically often available in, if you have something that's new and restricted, that might be available only in those colors. to really consider immune population biology and resolution when building a broad versus deep immune panel. So to step back about what that means, for instance, if you're building something and you have an incredibly rare population and need a certain amount of brightness, you really might need to take out a certain number of fluors to get as good of a detection as you want, because you will start to lose some of your resolution as you build to more colors. So that's important to really think about. Also thinking about matching that brightness if it's really a dim population. And then, thinking about in a deep panel, you're going to really be dealing a lot more with dye performance than a broad panel where you could position, for instance, antibodies that would only be expressed on B cells or T cells or antigen presenting cells, for instance, to avoid those more problematic dye combinations. Problematic dye combinations are not always identified with just the similarity index and the complexity score. So as I pointed out, the similarity index will identify something pairwise that's happening, and the complexity score will tell you overall, how does this overall panel compare to something else, but of a similar size. But it doesn't necessarily capture, for instance, if you do have this spectral bunching. And so just being aware to kind of think about, have I put 3 very similar dyes in a similar space in the panel, and maybe I want to avoid that. After you've built your panel, titrated, really thought about the populations you need to resolve, to identify and build around problematic perpetrator dyes and reduce spectral victims using the spectral spread matrix. So really looking at that row and column sum, and it's a relative score. So the idea is, and you could do this just as well on an Attune versus an Aurora in terms of, a conventional or spectral panel. But you really need to sort of be thinking about where are the places where, unfortunately, especially in a spectral panel, this might not be the best way that you've built that. And in the end, it's not about how many colors you have in your panel, but the resolution of the underlying biology that matters. And so that's really important to keep in mind. You could build 40 colors, but if you don't have resolution of the populations that you need, then it doesn't matter, because that's going to affect your ability to publish and get, an understanding of the data that you really need. So keeping that in mind as you build to that. And so with that, I want to thank everyone for their attention, and I'll be happy to take any questions.
Thank you, Seddon, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question today. What is the size of the phyton?
Yeah, so the phyton is about 20 nanometers if you measure across the cruciform structure. It's about 150 kilodaltons. So just to give you some kind of measure, PE is often, I think, in the 220 range. So about similar in size to that.
Thank you. Question 2, is cell blocks an FC block?
Yeah, really good question. I think there's a lot still to be understood about different blocking reagents. So cell blocks is not an FC block. Cell blocks is required with all NovaFluors, and what it does is prevent bad interactions between dyes and cells. And so, this would give you additional background. I would recommend adding that into your antibody mixture because it will give you, the best blocking, as well as you can add, either the brilliant or the super bright buffer. So those are actually blocking dye-dye interactions for the polymer dyes. And so you can actually use FC block, a brilliant or super bright buffer block, and the cell blocks all together to prevent any, basically non-specific binding that you may run into, but they do work in 3 different ways.
Very good, thank you. Next question, can I get a 1-to-1 labeling using a conjugation kit?
So yeah, really good question. I didn't go into that so much today, but what we have is The conjugation kits work by attaching an oligo to the antibody, and then basically that oligo antibody can be annealed to the NovaFluor of interest. And so that is a 1-to-1 ratio. However, with the conjugation kits, the difference is that we are purifying when we sell our antibodies and get that 1-to-1 labeling. We are actually purifying out excess dye. And so with the conjugation kits, you might be adding more dye into that mixture, so you don't fully get that 1-to-1 limit versus, say, getting that purified.
Okay, well, that brings us to our next question. Are NovaFluor's tandem dyes?
Yeah, really good question, and slightly complicated answer. So they are tandem dyes in that they use basically fret, so a donor and acceptor molecule to help transfer that signal. And so we typically think about tandem dyes as having that. The difference, however, tandem dyes are also is a term used to indicate the fact that something like PE size 7, you can't really control chemically how much size 7 you have. And so in flow cytometry, tandem dyes have been used with the idea that, okay, if you have more size 7s on this particular PE versus another conjugate, you would really need that specific lot because it would affect the signature that you have. So the difference here, we try to think about NovaFluors as being an entirely different category of dyes. And the reason for that is that the signature is the same, and that's because we're positioning the donor and the acceptor at precise positions. And so it's going to be the same, every time in terms of those positions of the dye.
Okay, thank you. Next question we have just came through. Can NovaFluors be used for applications outside of flow?
Yeah, so we're definitely working on that. And I think I would say there's more to come. We're certainly looking at things like microscopy. So I'd say keep watching the space for that.
Okay. And does buffer composition impact NF fluorescence?
So when you think about the NovaFluor fluorescence, there's no real quenching per se. So we've looked at fixing this with different dyes, looked at different NovaFluors and then different fixation procedures. So none of the buffers really seem to affect the NovaFluor. So for instance, you might be thinking about, for instance, super bright and brilliant dyes where they have that interaction, and sometimes there are issues with using that blocking solution that affect that. There's really no issue for that with the NovaFluors. There is some, I think when you fix with paraformaldehyde, there are differences in a number of, in some fluors in terms of that staining index. But we really don't notice anything that's specific to NovaFluors. That's just true for a number of dyes.
Yeah, it looks like we're nearing the end here. We have time for just one more question. This final question is, can cell blocks be used with dyes other than NovaFluors?
Yes, and it will actually help, especially with tandem dyes and non-specific binding. So for instance, monocytes will tend to sometimes take up tandem dyes a little bit, and so you'll see more background. And so it performs very similarly to, say, a true stain monocyte blocking buffer, in that it will prevent some of those non-specific binding to the tandem dyes. So it will help overall with other fluors in your panel.
Terrific. Well, thank you, Seddon. Before we wrap up, do you have any final comments for our audience today?
No, just want to thank everyone for attending, and please let me or the team know if there's any additional questions. We're certainly happy to follow up with you on that.
Okay, terrific. Well, thanks again, Seddon, for your time today and your presentation. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thanks.